Good morning, good afternoon, good evening, wherever you are. Welcome to Virtual Grand Rounds. Premier education brought to you at no cost, thanks to our sponsors, Medtronic, AstraZeneca, and our affiliate partners, Medela, the Science of Care. I'm Eric Lim, and this week it's a great delight for me to present Professor Anne-Marie Dingemans from the Erasmus Cancer Institute to talk to us about really important changes of potential horizon scanning, or what may be important for multimodality treatment for surgeons, on her topic today entitled Antibody-Drug Conjugates for Lung Cancer. Anne-Marie, over to you.
Thank you very much for the kind introduction and inviting me, Eric. So today I will discuss with you the antibody-drug conjugates for lung cancer. Here you see my disclosures. So I will discuss first the mode of action. So here you see an antibody-drug conjugate, which consists of an antibody, which of course attached to a target on a cancer cell. Then via a linker, you have a cytotoxic or an immunostimulant, which is called the payload. So this is seen as a kind of Trojan horse. So you really directly deliver the payload at the sites you want to have it active. But I think we have to understand that the payload is mainly chemotherapy. So it's not a new drug, different type of cytotoxic agent. So how does this work? We have the antibody-drug conjugates. It binds to the antigen. Then you have internalization. You have release of the payload into the cancer cell. And by this, it has an interaction with the target, which can be the DNA, the microtubules, or most times topoisomerase 1. So the same mode of action as we see with chemotherapy. But there's a bystander effect as well, because only a fraction of this payload is released in the tumor or in the tumor cells. And thereby it's important that there is some extracellular release where this has activity as well. This is the bystander effect, but it can also lead to toxicity, of course. So what do we know till now of ADCs and lung cancer? Here you see an overview of ADCs in development, which is already is not updated because there are many more ADCs in development currently in lung cancer. But I will show you some results of these ADCs. So we have trastuzumab deruxtecan. This is approved in EMA and FDA approved. And we have also some ADCs which are already halted because the phase 3 trials failed. So antibody-drug conjugates are there already for a very long time. And I think when we want to use an antibody-drug conjugate, probably we want to select a patient for the tumor target antigen. So with this, let's go to HER2. We know that HER2 mutations are actionable genomic alterations in lung cancer. It's rare, it's only 2% of the patients. And the DESTINY-Lung02 study showed that with trastuzumab deruxtecan in HER2 mutated non-small cell lung cancer, you see it was a single-arm study, a waterfall plot which really goes down, so nearly all patients had benefit, and the overall response rate was around 50%. Probably that's not as high as you expect in target-delivered chemotherapy, because all these patients had HER2 mutated lung cancer. However, the duration of response was really long, 16.8 months. So efficacy in around 50% of selected patients with long-term durations of response. A randomized phase 3 study in this setting is ongoing. So a very rare population, but a randomized study where T-DXd is compared with standard chemotherapy. We know next to this that HER2 overexpression occurs as well in non-small cell lung cancer. And probably because it is binding of the antigen, or the antibody to an antigen, HER2 overexpression is important as well as target. This is also the DESTINY-Lung01 study. And there you see that the overall response rate is at 34% and not really dependent on the level of HER2 overexpression. The question is, do we need a biomarker? And what type of biomarker do we need? Because it's shown that probably HER2 ubiquination and internalization is more important than the overexpression. So this might be that the biomarker on the outside of the cell will not reflect actually the activity of the antibody-drug conjugate. Let's go to another biomarker, another ADC. This is c-Met. We know that c-Met is overexpressed as well frequently in lung cancer. We know that c-Met overexpression is a resistant mechanism for patients treated with osimertinib in EGFR-mutated non-small cell lung cancer. And we are not sure whether c-Met overexpression is really a driver of cancer, but probably that doesn't matter when you study ADCs. In Teliso-V, the luminosity studies, Teliso-V is studied in several cohorts. In EGFR wild-type cohorts, where it's selected based on c-Met high or c-Met intermediate expression, so immunohistochemistry, and also in patients treated with osimertinib, so EGFR-mutated patients, where also there are cohorts based on c-Met expression. What we see in the patients with wild-type non-small cell lung cancer, we see overall response rate around 22%, and the response rates seem to be a bit higher in the c-Met high patients, so high c-Met expression. Next to this, Teliso-V is also studied in combination with EGFR, with osimertinib in patients with EGFR-mutated non-small cell lung cancer after osimertinib failure. What we see in here is a small cohort, but I think interesting overall response rate around 58%, which I think is very promising. Teliso-V is also studied in a randomized control study in the second line versus docetaxel in patients with EGFR wild-type but c-Met overexpressing non-small cell lung cancer. So in this phase 3 study, they already studied Teliso-V based on c-Met overexpression. The question is whether this is the right biomarker, as I showed you that probably the internalization is more important. So the question is whether this study, when it reads out at that time, whether this is already the best biomarker for this ADC. Then we come to our next ADC in lung cancer, and that is patritumab deruxtecan. It's based on HER3 overexpression. This is a study performed in patients with after failure of a TKI. And what we see here is really deep responses. So we see that some of these patients have complete responses, and probably that is what you hope for with these ADCs. But the duration of response is around 6.4 months, but half of the patients have more than 6 months duration of response, which is, I think, of interest as well. So overall response rate around 30%, but the responses seems to be durable. And here, this is also interesting. I think more of this translational research, which is so important in this setting, it shows that osimertinib increases the HER3 expression, but probably also the internalization of the HER3 DXd. So the internalization seems to be much more important than the expression on the outside of the cell membrane. And this is studied as well in biomarker unselected patients in a randomized setting. Then we come to TROP2, and TROP2 in this setting, there we have randomized phase 3 studies. And why do we have randomized phase 3 studies? Because TROP2 is expressed in nearly all non-small cell lung cancer, so here the studies are performed in an unselected patient population. So there are two important studies, one with Dato-DXd and the other with savolitinib deruxtecan, and they are both in the second line setting comparing this against a docetaxel. And we know none of us like docetaxel in the second line, but we also know it seems to be hard to improve outcomes above a docetaxel. And these studies have more or less the similar outcomes. So what you see with Dato-DXd, there is a bit of improvement of progression-free survival, but overall survival is completely the same. And I think these results is very, very disappointing. This is not what we expected from these ADCs in the setting of non-small cell lung cancer. The toxicities are different, however, so comparable outcomes, but different toxicities. And this has to do with the difference of DAR, so meaning the drug antibody ratio, which is higher for sacituzumab govitecan, and thereby leading to higher rate of toxicities, especially diarrhea. And what you see is that the toxicities are chemotherapy-related toxicities. And then there was an interesting analysis presented by Marina Carrasino at World Lung last year, and that is about the biomarker, because we know that the efficacy is not dependent, or the immunohistochemistry expression of TROP2 doesn't predict the efficacy of the compound. And here it's it's an interesting test. It's about TROP2 normalization membrane ratio, and it is shown that when this biomarker is highly expressed, that these patients seem to have more benefit. So the biomarker development in the setting of ADCs is much more complex than expected in the beginning, but seems to be very, very important. Then there seems to be much more activity of Dato-DXd in patients with driver mutations, which is a bit difficult to understand why this is. But this is studied in further studies, and here you see the overall response rates of these patients in this setting. What have we learned of all these years of ADCs? Well, we know that curative chemotherapy comes from multi-agent combinations, and we know that ADCs, the payload is chemotherapy. So many studies are now ongoing, combining ADCs with other chemotherapy or with immunotherapy. And these are all very studies in early development. Let's go to the toxicity, because we think when an ADC delivers the chemotherapy, the payload, really at the tumor, probably you expect less toxicity. But this is not the case. And therefore, it's good to understand how ADCs work and what are the important components of the ADC. So one important compound of the ADC is, of course, the payload. That's the chemotherapy. And we see that the toxicities in all these ADCs are comparable to the chemotherapy. So with the topoisomerase 1 inhibitors, we see diarrhea and neutropenia. And with the microtubule inhibitors, we see toxicities comparable to the taxanes, as we know. Then it's important to understand, then it's important to look at the payload potency. And we know how the higher the drug to antibody ratio is, the more the toxicity is there. This toxicity can also be on target, but off tumor sites. So when you have HER2 expression or TROP2 expression on other sites than the tumor, we will deliver the chemotherapy, the payload as well. So therefore we know that with HER2-related ADCs, we see cardiotoxicity. With TROP2 ADCs, we see rash and mucositis, and well, you see the other things here as well. Next to this, these binding of the Fc receptors can bind to the immune cells, which give off-target toxicities as well. And probably the pneumonitis can be explained binding on this type of toxicity. And the pneumonitis is something we see with the trastuzumab deruxtecan. Next to this, an important compound of the ADC is the linker stability. And the more cleavability of the linker there is, the more release will there be outside of the tumor cell, and the more chemotherapy-like toxicity is observed. So where are we with all these years of ADC development? I think the technology is there, and the technology improves the ADCs enormously. And there's a lot of knowledge in the last year in the ADCs. And I think the ADCs are there to stay. But we are still awaiting important outcomes in lung cancer, because I think till now the randomized trials in lung cancer are a bit disappointing. But with the improved understanding and improved technology, I hope the outcomes will improve as well. So the take-home message, I think the ADCs are the new kids on the block. And when we have new kids on the block, we have to understand them as clinicians. We have to recognize, therefore, the unique side effects of each ADC, which, as explained to you, has to do with all the compounds of the ADC. We need to educate our colleagues, ourselves, our nurses on the ADC toxicity. And we can have off-target, off-tumor toxicity, on-target, off-tumor toxicity, and target and payload-independent toxicity. ADCs, it's not really there for the clinical practice. I think only we have DXd for the HER2 mutated patients, And many, many trials are ongoing, and we have to await these results. And with this, I'd like to thank you for your attention.
Thank you very much, Anne-Marie, for a fantastic overview of ADCs for surgeons. Just going to bring you a few steps back. Can you maybe describe to us how ADCs are being administered? Presumably they're intravenous, and what the actual schedule is like, and how long in general these systemic treatments are given for?
Yeah. It is an intravenous administration, and depends the schedule. It's a bit like chemotherapy, and most of them till now are prescribed till progression. So meaning that you can have cumulative toxicity comparable to the chemotherapy as well. So when it's a microtubule payload, you really will have neuropathy as well. And I think, so we have to understand it's chemotherapy, and as a lot of off-target delivery. So the toxicity, so we have seen with Dato-DXd, we have seen a lot of mucositis. And of course, because we learn these toxicities, and there are a lot of preventive measures, you see over time in the clinical trials that the incidence of the severe toxicity really decreases, as the same as with the pneumonitis, because with recognizing early the pneumonitis and giving treatment for this, the severity of the toxicity really decreases.
So increasingly, we're seeing this because as surgeons, we're very used to the concept of 4 to 6 cycles of systemic treatment either before or after. And then suddenly this year with the publication of all the multimodality immunotherapy studies, we're now looking onto 1 year of treatment. And with ADCs, what you're saying is that the potential future landscape for systemic treatments could be, as long as it takes until progression happens.
Yeah, but that's then that's indeed in the stage 4 setting. I think all compounds are first developed in the metastatic setting, and when they have shown improved outcome there, and next to this safe toxicity profile, it will be going to the earlier setting. But I think first we need really solid data in the advanced lung cancer setting.
Yeah. I mean, it's interesting that you've mentioned that because the relevance of this talk to surgeons is that AstraZeneca is now already launching Tropion-Lung-12, evaluating ADCs, in the resectable lung cancer setting. And that's why I think surgeons need to be, and I think that's probably the first company, but many other companies will be following suit.
Yeah. So I think when you go to the surgical, to this setting, the importance is that probably you can better have the biomarker development, which seems to be, in my opinion at least, very important, because we see that not all patients respond. So for the TROP2, we know that the TROP2 expression is very high. It's independent of the immunohistochemistry doesn't predict, but not all patients benefit. So I think with an ADC, probably we want to go to a selection. And then when you go to the surgical setting, of course, because of the specimens you have available, probably you have more possibility to come to selection. But I think for surgeons, it is important and to know it is still chemotherapy. So you can still have thrombopenia, neutropenia, anemia, all these things happen there as well.
And that's so relevant or pertinent to surgeons because some surgeons actually do their own follow-up for their patients going long term. And we were also having a discussion about this, whether or not that's appropriate, because as surgeons in the good old days used to follow their patients up to 5 years, you're going to start to see complications from immunotherapy, from new agents, so on and so forth, that I don't think surgeons are equipped to deal with. And at some point, as an oncology community, we're going to have to decide who the appropriate or what the appropriate method is for patients follow-up. And I can see patients with systemic treatments to be oncology followed up, patients without systemic treatment, either surgeon or chest physician. How's the follow-up in the Netherlands?
Well, in the Netherlands, I think we have a different situation because in the Netherlands, the pulmonologists treat the patients with chemotherapy. So the patients are not seen by medical oncologists. And as the pulmonologist also, see these patients at diagnosis, of course, the pulmonologist is always in the lead. So the patients are not followed by surgeons and not by radiation oncologists. They are always followed by pulmonologists.
I think that's very, very sensible. I think a lot in the UK, my MDT as well, followed up by pulmonologists.
Yeah.
Anne-Marie, you alluded in your talk that, ADCs may not be delivering the promise when it first came out. It's going to be such an amazing drugs. Um, can you go a little bit more into that? In that, what were your expectations when the drug compounds were released, and going forward, do you see this improving or not?
Yeah, so I think what we have seen, well, the lung cancer field, of course, has completely changed by immunotherapy, where we have this. I think we can say we cure patients with stage 4 disease with immunotherapy. Not all, but we cure. We have patients with 5 to 10 years without any progression of disease. And next to this, we had these small molecules, these tyrosine kinase inhibitors. So, and I think with these tyrosine kinase inhibitors, we see 80% of patients having a response. We see that more than 50% of patients is alive after 5 years. So I think what I at least expected from ADCs is when you have biomarker selected delivery, I hoped to see this very, very high response rate. And then you see with the trastuzumab deruxtecan in the patient with HER2 mutations, we see these response rates are around 40–50%. Probably this is higher than with the chemotherapy, but it's not the 80% which we see with the TKIs. And that is, well, probably our expectations were just too high. And I think now we have to prove that it is better than chemotherapy, because in these randomized phase 3 trials in the second line setting, compared to docetaxel, which we do not like at all, doesn't show any benefit. So I think that, but what we see, and there are so many trials in development, so it's not of interest for these type of presentations, of course, but the technology is really evolving. So what I hope with evolving technology and with bispecific ADCs and all these type of things, we will have better outcome as well. So probably our expectations were too high, but I think at least we thought for these top 2 ADCs, we all thought that the outcome would be better than docetaxel, of course, which was not the case at all.
Understand. Thank you. Question from Dr. Ballock, who's asking about costs of ADC compared to chemotherapy in general.
Yeah, it's very high, of course. It is new. It is completely new, and all these chemotherapies we have are out of patent. So pemetrexed was a very expensive chemotherapy as well, but the cost of chemotherapy are currently very, very low. So I think we need really, at least for the Netherlands, we need a very good benefit. So increase outcome before reimbursement will be there. And especially reimbursement in all countries over the world, of course. Yeah. So the schedule, so now the schedule is still in the stage 4 setting till progression. Probably we'll have to look at the schedule as well. Do we need more cycles, or is 4 cycles just enough? But these are all things we have to study. Yeah.
Dr. Okonta is asking, we tend to go down one single pathway, in general, TKI or immunotherapy or ADCs. What are the future of combination with, say, checkpoint inhibitors and ADCs and multi-variation combinations that you might think about in the future?
Well, I think it's not probably the future. All these trials are ongoing with all kinds of combinations, especially, of course, with the immunotherapy, because we know that, although we have patients with long-term benefit, there's still an important group of patients who do not benefit at all. So their combinations are ongoing. And there are these B-specific ADCs also targeting immune cells as well. So there are a lot of ADCs in development, but also in combinations with other treatment, and thereby replacing regular chemotherapy. But I show you that toxicity can be even more severe than with the regular chemotherapy. So I'm not sure whether it will replace chemotherapy very easily.
The Balog is also asking, how soon will the side effects happen, just in the future for surgeons? For example, if they're given before surgery or after surgery, when can we expect patients to develop these side effects?
Very early, because it's exactly the same as chemotherapy. So you have to check for every cycle the hematological toxicity, of course, and the mucositis can be there already in a few weeks. So the toxicity is very, very rapidly. Interestingly, with TXDXD, trastuzumab deruxtecan, you see pneumonitis, and you see that it's, I didn't show you the curves, but it is already in the first month. So it's not really a late toxicity. But what all these studies show that when you have early preventive measures, you can really decrease these toxicities. So I think when you have ADCs in a neoadjuvant setting, you have to be careful for all chemotherapy-related toxicities.
Yeah, most of our surgical guidelines tend to focus on how to make patients better, how to recover quicker. But I think that the next generation of surgical guidelines, we really need to think about pre-operative evaluation with respect to all these systemic treatments. These are all going to be like the new area for surgeons. We're going to definitely be involved in the management, in the perioperative management of potential complications that can happen, and when to operate. Because in the past, it was just neoadjuvant chemo or adjuvant chemo, and the neoadjuvant chemo with the complications are relatively not too bad to manage. But this is a whole new horizon for us. Dr. Addy is asking, are there any contraindications in terms of comorbidities for ADCs at the moment, in general?
No, not in general, and it's exactly as the payload is, so the chemotherapy. So I think for the pneumonitis, we know that patients with interstitial lung disease, you will not treat them with trastuzumab deruxtecan. So you have to know it for every ADC, and I think this is. Well, of course, we had time to learn side effects from every chemotherapy, but now there are so many ADCs in development, and they all have their own toxicity profile. So I think when we have an ADC which is approved, we really need to understand how this ADC works and what part of the ADC makes the toxicity. And with this, we can have preventive measures, of course, and guide also our patients. So every ADC has its own profile of toxicity.
Going forward, centers, specialist centers like the Brompton, where we only do heart and lung surgery, we don't actually have on-site oncology. I'm just wondering how long that can go on for, because now treatments are so closely integrated, including the complications, so on and so forth. Standalone specialist, thoracic surgical centers, I think may become, it may need to evolve with it or otherwise be phased out, because surgeons won't be able to manage these complications on their own. So it's a very, very interesting development.
Yeah, so I think because the neoadjuvant chemo IO, of course, has completely changed the world. And this, I think, will be really the future, that we are going to give neoadjuvant treatment probably to all patients with lung cancer, isn't it? Because you have the idea that with this complete pathology response, of course, we cannot predict on forehand who these patients are, but these patients have really a very, very good outcome. And you can think about that. Probably it's important with immunotherapy that the cancer is still there to really have the good immune response. So indeed, I think that the interaction with medical oncologists or pulmonologists and surgeons is becoming more and more important, because nearly all patients will have some combination therapy.
Excellent. As always, there's way too many questions than we have time for answers. So thank you so much. Next week, we're going to talk a little bit about new targeted treatments on HER2 and RET with Justin Gaynor from Mass General, which leads me once again to thank our sponsors, Medtronic, AstraZeneca, and Medilia Cyclecare, and of course, especially to you, Anne-Marie, for explaining it to us in such an easy to understand manner and, continue to develop the field as you have been doing. Thank you so much.
Okay, thank you.
Have a good day. Bye-bye.
Bye.